So, good day. My name is Janice Macayan from BSN 1A, and today we will talk all about the controlling microbial growth in vitro. So, it means that we're going to focus on the microorganism that lives or usually located sa environment or outside in our body. So, there are factors affecting growth. So, first is the uh, availability of nutrients. So, nutrients are very important to to an bacteria, viruses, or fungi because it is it is one of the factor that make them that they use for them to replicate and um, to that will make them survive for their everyday um, living. So the next is the moisture. So moisture is also very important in microorganism because this is where moist moisture surface is where they like because um, it is where they multiply, they replicate. So especially when that specific surface that is good in temperature and lot of nutrients. So the next is the temperature. So yeah, temperature because um, bacteria, in order them to survive, it should have be a neutral temperature for them. It depends on what kind of microorganism are they. So the next is the pH. So the acidity or the alkalinity of the environment, or yeah, where the is microorganism are present. So so it is for the microbial growth. It is good if it's neutral or just what the specific specific temperature that microbes need. So um, for examples, let's say archaeans. Archaeans are microorganisms that lives mostly in hot environment, so they can survive there, and some are not. So here's are we call thermophiles. So they are microorganism grows best at high temperature. So those microorganisms that usually live or can survive in hot in super hot temperatures are called thermophiles. Or so mesophiles. Mesophiles are microbes that grow both moderate temperature. So mesophiles, microorganism luwati nga can survive in a moderate temperature laliwat. Example sini it ada mga plants, animals, and hot soils. Or kung hatawo and mga aton indigenous microflora. And psychrophiles liwat are microorganisms that prefer the cold temperature. So, siya iniraan, gustuhan mahagkot nga temperature. They can live there. pH na kita, di ba? Back to pH. pH liwat ba dahil tagin si saring nga acidophilic and alkaphilic. So, balik So an acidophilic and microorganism in that usually able to survive in a acidic environment, especially um, in the stomach of a person, such that. And while the al alcoholophiles prefer alkaline environment. So these are microorganisms that can survive in high in the presence of high alkaline and alkaline. Examples in are choleras. Yeah. So the next is osmotic pressure. So osmotic pressure, this is the pressure that is exerted on a membrane by solution both inside and outside the cell. So yung osmotic pressure amin yan din sa nga from higher to from lower from higher to lower concentration, from highest to from highest to lowest, from lower to highest concentration. To malinis sa nga hypertonic solution is suspended. Asya na gets setting for example, and cells kung damot water hasulod, tas kuti ay it salt, tas kuti ay salt, asya na ba? Kung kuti ay water, I mean kung ano ba ito, presence of salt hasulod tapos kung sugad it usa nga cell kung damoy tubig tas kutay salt tagawa kuan and pressure hini para magpantay magawasan water nga to hagawas. So kung in opposite liwat kung kutay laluwat and salt ta sulod tas damon water gawat, so may liwat is masulod liwat and water nga to hasulod. So from higher to lower concentration para balance. So the next is the barometric pressure. So I'm familiar with barometric pressure, but it is most bacteria are not affected by barometric pressure, just a minor change lang. Examples nila mga barophiles daw na microorganism. These are microorganisms that usually live in the deep oceans. So the next is the gaseous atmosphere. So these are microorganisms vary with respect to the type of gaseous atmosphere that they require. So ini yung mga microorganism can live with oxygen and can live without oxygen, or we call the anaerobe microorganism anaerobe microorganism. So usually humans eat ang tawag ito, room air is 21%. So, depende on kung ano ang microorganism. Di meron prefer. So, next is encouraging the growth of microorganism in vitro. So, we have this culturing in the lab. So, we have different kind of culturing in the lab. The first one is the bacterial growth, wherein we have different medium that uses wherein it is where bacteria or microorganism being cultured in order to identify if that microorganism can live and what are those characteristics of that specific microorganism. So we have called the solid medium. So it is a where the binary fission continues. So, and binary fission, so dila ang solid medium bidaan ng multiply and microorganism. So, we have the bacterial colony. Bacterial colony liwat, wherein didahan ang tawagin nihan solid medium and colony is an pundok kan microorganism nga ada ito nga medium. Yeah. Then the next is the culture media. So the culture media, it is an artificial media usually in laboratories. So that is, it is where it is culture media or medium are used by mga pathologists for culturing a specific specimen in order to identify what are microorganism present in that specimen and to identify the what kind of treatment should or what to identify the characteristic of that microorganism present in the specific specimen in order to give a specific and accurate antibiotic for the specific complaint or disease. So we have the first category in the media. We called chemically defined. So chemically defined media. So in here, it's a type of culture media wherein media, it is where it is classified based on whether the exact contents of the media. So it is used by adding a certain number of grams of each of components such as carbohydrates, amino acids, and salt. So, kumbaga, ang media, yung bumutangan hin, carbohydrates, either carbohydrates, amino acids, or salts. So, ganito. So, next is the complex medium. So, ini yung complex medium liwat, used in which the exact contents are not known. So, contain ground of digested extract from animal organ, fish, yeast, and plant which provide the necessary nutrients, vitamins, and minerals. So, ini yung complex medium. So, bagat diri ko din kung bisla itada hit sulod hit medium. So, for example, ini kanan animal enzymes, so hinubutang yang to in order kung ano't mangyayari hit to sa may bacteria kung present sa tirangan to. So, and next is enriched medium. So, ini nga enriched medium, from the word itself, enrich, to ini, para ini usually mga fastidious bacteria or microorganism and mga magkaon nga mga bacteria, so that yun bumutangan and usually gagamit ini is blood agar or an chocolate agar. Sige. And selective medium liwat ini na culture media is selective iya. So kung ano la iya anong tawag ito, kumbaga as added inhibitors that discourages the growth of contained organism without inhibiting growth of organism being set. So may daniya inhibitors nga to, pero kung anon and kailangan nga microorganism diri hiya bagat diri gini stop ang iya growing mechanism. So, for example, sinin makangki agar. So, in pinihiya nga medium inin medania o nag-inhibitiyahin microorganism and gratan and organism pero except han can be and again angin kung ano man and makuan. So, next is the differential medium. So, ini permits the differentiation of organisms that grow on the medium. So, example is the macaque agars frequently used to differentiate among various gram-negative bacilli that are isolated from fecal specimen. So, for an gram-negative bacterium able to ferment lactose produce fecal, tapos unable. So, for example, in a differential, so, for example, ang gram-negative, so, kung ang gram-negative, nakaka-ferment yan lactose o nakaka-breakdown yan lactose, nagpipink and part and colony. Pero ang diri nga ni nakaka-ferment is kuha niya, waray layan niya color. It's so good and purpose and differential medium. So, amulat. So, next is the inoculation of culture media. So, pag inoculate it, culture media is dapat routinely. So, dapat may schedule niya. So, kumbaga ini dapat routine. So, the next is the importance of using a sterile technique. So, what is the importance of using a sterile technique? So, it's very important to use sterile technique because this is for microbial, for in studying microorganism. So, it is important sterile technique because this exclude all microorganisms from particular area. So whatever we do in, for example, in a hospital, especially when we do a aseptic technique, we should always perform a sterile technique because that procedure needs absence of microorganisms. So everyone who will perform should be careful not to be exposed in a contaminated area. So this will exclude all microorganisms from a particular area. So next is the incubation of microorganisms. So may kita 3 types of incubators: a carbon incubator, a non-carbon incubator, and a anaerobic incubator. So ini nga a carbon incubation. So to each cylinder of carbon dioxide attached. So into incubator to maintain a carbon dioxide concentration about a 5 to 10. So ini asya niyan mga microorganism that live in they are bisan carbon dioxide ini, they are not an anaerobic microorganism, so they can live in aerobic organisms. So usually it carbon dioxide ini is 5 to 10%. So examples ini ang mga capnophiles. So asya to. So next is the non-carbon dioxide incubator. So it is an incubator containing room air 20 to 21%. So for non-carbon dioxide, so ang room air line niya. So kumbaga kunan la 20 to 21% asya man talaga. So anaerobic incubation is containing atmosphere devoid of oxygen So, ini nga katulungan an anaerobic incubator. So, anaerobic, so without presence of oxygen. So, asya. So, these are organisms that, this is to identify if that specific cultured specimen or bacteria are able, in incubation are able to live in an anaerobic or room air temperature, or yeah, um, uh, environment. So, so the next is the population counts. So there are two types wherein bacterial population counted. So the first is the spectrophotometer and the variable plate count. So an spectrophotometer is where a beam of light pass through a liquid. So it is determined the total number of bacterial cells in a liquid. So kumbaga, it is light lahiya nga gintutungod ha liquid tungod nga may presence in microorganism. Tapos so asya na ito giniap nila kung pera and presence and total number and bacterial cells presence in that specific area. So sunod liwat and variable plate count, used to determine the number of viable bacteria. Determine the number of viable cells. So, what that to. So, usually, serial dilution are prepared. Yeah. Then, overnight, the number of colonies counted. So, serial dilution, siya ito ni bubutang. Tapos, after inoculation, kakantuhan an agar, tapos after a day or depende, tas siya ng colonies are counted. So the next is the bacterial growth of a bacterial population growth curve. So there are 4 phases in the bacterial population growth curve. So the first is the log phase, the logarithmic phase, the stationary phase, and the dot phase. So we're gonna draw it so that we are going to imagine it. So the log So, and log phase, it is where absorbs nutrients, synthesized, and with enzymes, and prepare for cell division. So, kung pala niya, nagpapakabuhi, charot. Nag-start pala hiya, nagpapalakas, bagat subat. So, kumbaga, for example, in log phase, adi pala hiya nga part. So, adi pala hiya. So, adi pala hiya nga part and log phase. So, pag kumadi nakita logarithmic phase or log phase, ano sa liwat logarithmic phase, nagmamultiply na niya rapidly, like nagdadamo ya dayon, tapos asya to na double and growth niya. So, mangyayari sa na So, tumigdaman niya, logarithmic nga ni, meaning nag-multiply, nagdami niya. So, adi na hiya dayon. Adi na hiya dayon yan. So, ang pagkadiliwa tayo stationary phase, nutrients are up and the concentrations of the toxic waste produced from the metabolizing bacteria build up. The rate diversion slows and the greatest population diversity. So, ngan hiliwat, asya na to, since damo man hira, so asya na to, so damo gyap and waste nga from tigangan era pag, ano ba ito, pag, era pag, ano ito, pag, may mga waste niya, so nagtulog so taas and nutrients since damo man hira, so nag-aalagaw gihapon, so dito, so kung pera na mamatay, kung pera buhay siya lahat na mamatay, so bagat steady line niya, steady lang siya, equal lang kung pera na kukuha na siya. So, ang that phase liwat, tungodhan overcrowding, tungodhan toxic waste nga presence nga danera, tapos activities to increase and the nutrient supply decrease. So, asya to. So, ang nangyayari tungod ang kadamo nera, overcrowding and nutrients pa nag-aragaw nera. So, amo na to mag-dead phase na. Kumbaga, in lag phase, adi nga number 1, adi ang logarithmic phase, adi stationary phase, tas adi ang dead phase nahan microorganism or an bacteria. So, next is the culturing obligate intracellular pathogen in the laboratory. So, these are microorganisms that can only survive and multiply within living cells. So, ini nga microorganism, usually they are viruses. They grow and inoculated into embryonated chicken eggs, lab animals, and cell culture. So, usually, nira, ginagamit nira or in order to be, anong tawag ito, to be studied, kailangan nira ikadahin sa nga living cell. So, example ito, and mga eggs, lab animals, and cell cultures. So, the next is culturing fungi in the lab. So, fungi, so, iniliwat, makuri mag-culture in fungi kay they are damot era spores. So, kumbaga nag-iiba hera kunwari yan na sugarit era kuan. So, pagka next nga pagkuan liwat nag-iiba hera. So, bagat basta po, so basta may daheragin si siling it mga kuan ng class 2 biologic safety cabinet kanin para fungi. So, ay mga infectious man ko din examples han and kogya na nga super makuri mabilnganit iya specific antibodies nga para malabanan ini nga virus. So, the next are culturing protozoa in the lab. So, usually mga amoeba, so it is somehow cause a serious infection of the central nervous system because infection that are difficult to diagnose by other methods. So, So the next is inhibiting the growth of microorganism in vitro. So kumbaga niliwat kung naanon pag slow down or pag stophan era pagdamo. So it pinaka first is sterilization. So what is sterilization? So it is the complete destruction of all microorganisms including cells, spores, and viruses. So accomplished by dry heat, autoclaving, gas, mic, various chemicals, types of radiation. So ini usually ginbubuhat through autoclaving, dry heat. So usually may nakita yung sisaling na disinfection, pasteurization, disinfectant, antiseptics, and sanitation. So an disinfection. So it's the destruction or the removal of pathogen from non-living objects by physical or chemical methods. So for examples in disinfection and pwede ang mga inanimate objects. So kung maugaw pwede usually ginlalaysol. So may dapagihapan sinisring ang pasteurization. It is the heating process to kill microbes in which the method of disinfecting. So ang pasteurization is a method of disinfecting. Usually ang mga milk saan ginbubuhatan na ginpapasteurize. So next is disinfectant. So disinfectants and mga chemical agent used to eliminate pathogens. The strong substances that can be used living tissue. So siya yung mga disinfectants na gagamit para to kill microorganisms. So antiseptics, it's a solution used to disinfect skin and other living tissue. So antiseptics such as alcohol. So pwede mga sa iyo. So the next is sanitation. So sanitation is the reduction of microbial population to levels considered safe by public health standards such as these applied to restaurants. So sanitation and proper sanitation bisan and proper disposal lahin mga waste mga tapos sanitation kitchens dapat sugat especially mga restaurant. So the next is the microbicidal agent. So microbicidal sides means killing. So there are types of agent used to kill microbes. So for examples are germicidal agents, biocidal agents, and microbicidal agents are used to kill microbes or microorganisms. They are disinfectants used to kill microorganisms. Okay, the next is bactericidal agent. So this it's kill bacteria. Pero diri mga may da bacteria nga may endospores. So sporicidal agents are the one kills bacteria that has endospores. So and fungicidal agents kill fungi, including those fungi with spores. Spores. Next is algicidals are agents or disinfectant used to kill algae. Virucidal are disinfectant used to kill viruses. Then pseudomonicidal agents kill species and mga Pseudomonas, Pseudomembranous, charot. And next is tuberculoidal agents. So these are disinfectants that used to kill Mycobacterium tuberculosis. So the next is the microbistatic agent. So may din considering na microstatic agent is a drug that inhibits growth and reproduction of microorganisms. So ini nga, it is a drug. So that is gini-inhibit niya or gini-slow down and growth and reproduction han usang microorganism. So, and sinis na nga various bacteriostatic agents. So, ini yung bacteriostatic agents, gini-inhibit sa yung process or inhibit the metabolism and reproduction of the bacteria. So, asyad to para di rin na kuan and bacteria. So, gini-inhibit niya and growth. So, mga drugs that inhibit the growth of a specific microorganism. So, ang lyophilization liwat, it is a process of freeze-drying or rapid freezing. Freeze-drying. So, kung bagat stop and growthan kuan microorganism bagat so that by and freeze niya. Pero diri niya pwede parahan mga immunocompromised kay kumbaga wala lagi niya gamit kay immunocompromised naman so bagat diri kaya itlawas ma-freeze niya so bagat so next is the sepsis, aseptic technique, antiseptics and antiseptic. So sepsis is it is the presence pathogen in the blood tissues, whereas sepsis, the presence of microorganism in the blood tissues or kundi in media. So an asepsis liwat, asepsis from the suffix a without, so these are without the presence of microorganisms or the absence. So an aseptic technique, so with doing a technique that without presence of microorganism. And antisepsis liwat, prevention of infection, so bagat staying away from microorganism. Tapos, an antiseptic technique liwat, use of antiseptic type of antiseptic. So, ini bagat under yan and kanan antiseptic technique. So, antiseptic bagat same gad lagi pero. So, the use of physical methods to inhibit the microbial growth. So, on physical methods na hindi nga process. For example, it's either physical or chemical or both, hospital or clinic lab. So, and pinaka first is in heat. So, it is most practical and inexpensive. So, usually kasi, within niya two factors, temperature and time, and thermal death point and thermal death time. So, on thermal death point, any particular species of microorganism in lowest temperature that will kill all organisms and standards pure culture within a specific period. So, ini thermal death point. So, medali niya specific temperature in order to kill the microorganism. Well, ang thermal death point liwat, it is the length of time necessary to sterilize a pure culture at a specific temperature. So, kumbaga, may length of time hiya kailangan para magkamatay ang microorganism with a specific temperature luwat and kailangan. So, asya, to, and kanan heat. So, naluwat ang sinisiring nga dry heat. So, ang dry heat liwat is baking thermostically controlled oven provides effective sterilization. So, ini usually kuan na ini incineration or unburning of microorganism. Effective in destroying contaminated. So, ang mga gingamit in specific na procedure nga sugad ang nagkada yung procedure nga may HEPA, sinunta may hepatitis and patient. So, in order to kill or in order nga mawala na at diri makatapon na itong microorganism, ang bubuhaton is dry heat nga procedure wherein it's a warning of ang mga materials na gingamit. So, the next is the moist heat, wherein heat applied in the presence of moisture, as in boiling or steaming, faster and effective than dry heat and can be accomplished at lower temperature. So, usually ini kuang kuno presence of heat la in a moisture wherein syempre diba and microorganism or an bacteria man. So, may protein, chemi man gyapera. So, pag na loose o pag super may the presence of heat na which is not under bagat kuanhan era kuan kung nataas man or kuan bagat na alter and function. So, syempre it couldn't to coagulate sa asyad. So, for example, hini siya na. Oh, siya. So, next, an autoclave. So, an autoclave, it's a large metal pressure cooker used to steam under pressure, destroying completely or microbial. So, usually, gigamit din mga hospitals in order mga gamit nga for surgical instruments, sterilization han mga water. So, autoclave it gagamit to. It's a big machine, already so wet. So, basta it's like a baking chemi. So, the next is the cold. The microorganisms are not killed, but their metabolic activities are slowed and inhibit their growth. So, ini yung process nga kanan physical methods. Di rin na mamatay yung microorganism, pero it inhibits the growth of microorganism. As yan yung examples kanina, slow freezing and rapid freezing. Wherein ini rapid freezing diriya pwede or ini nga freezing method diriya pwede mga immunocompromised nga patient. Desiccation and pag-desiccate. Tapos radiation liwat from the sun, so and light rays so do not penetrate the walls but the cells cause damage to DNA. So asyan to. Tapos mga ultrasonic waves, so use means of cleaning and sterilizing delicate equipment. So gagamit gyapon yan. So the next is the filtration. It is used to filter or separate cells. So filtration, filter yan cells. So next is the gaseous atmosphere. So it inhibit the growth of microorganisms by altering the atmosphere in which they are located. So gina-alter and presence and gas. Gusto kunwari aerobes microorganism. So tawag ito ang presence and gaseous atmosphere. So gini-iba in order to kill that microorganism. So using chemical agents to inhibit microorganisms. So asya to kanina disinfectants, ang mga chemical agents here to inhibit the growth of microorganism. It's either temporarily or permanently. Pero may factors kita that affect the efficiency of the so on time, temperature, concentration, presence of protein in feces, bladder pass, types of microbes, number of microbes or presence of the spore. So safe explanatory man niya. Next is antiseptics. An antiseptic, just like kanina, that's used safely in human tissues, reduces the number of microorganisms, mga alcohols, alcohol to disinfect or kuhanan aton tissue panit para away from bacteria. So inhibiting the growth of pathogens in our kitchen. So usually aton syempre kuan manggod prone na natin ang kitchen microorganism kay tungod din mga aton gimpapalit gawas. So ganun mga gulay, mga kuan, so it is one of the most contaminated area of the house. So usually asya itong gigagamit ato mga disinfectant, antiseptic in order to kill microorganism presence in our house, especially in the kitchen. So that's all. Thank you.